All right, dual credit biology students, we're going to get into a little bit of detail here. So we're going to start stepping through these three major steps or portions of cellular respiration. The first one is called glycolysis. Glycolysis basically means splitting sugar or breaking sugars. That's exactly what we're doing. So concept 9.2 says glycolysis harvests chemical energy by oxidizing glucose to pyruvate. So what we're doing is we're taking a glucose molecule and we're splitting it in half. Glucose is a 6-carbon sugar molecule, and we're going to break that into two 3-carbon molecules called pyruvate. So we're going to oxidize the glucose, which means we're going to remove some electrons from it, and it's also going to be broken into two smaller molecules called pyruvate. This happens outside of the mitochondria. This occurs in the cytoplasm of the cell, and it basically has two stages. There's an energy investment phase, where the cell actually has to expend energy to make the first part happen. And then there's what you would call an energy payoff phase, which is where energy is received back from the final reaction or from the final stage of this. Glycolysis happens whether oxygen is there or not, so this does not need oxygen to occur. And so that means glycolysis can occur in environments that have no oxygen or with living creatures that live in environments that are deprived of oxygen. If oxygen is present, we would move from glycolysis on to other stages where we would oxidize the pyruvate, we'd go on to the citric acid cycle, and then to oxidative phosphorylation. So this can happen, this first stage, glycolysis, can happen whether there's oxygen or not, but it does require oxygen for us to move forward after this first stage. This little flowchart or model kind of helps you understand what's happening in glycolysis. So we're starting here at the top. We have what's called the energy investment phase. So the cell has to actually use energy to get this to happen. The cell actually uses two ATP molecules to break the glucose down into two pyruvate molecules. And that's going to also produce a couple of other things, two ADP molecules and two phosphorus groups. Using some ADP and phosphorus groups that are already present in the cytoplasm, then we get to the energy payoff phase where we have 4 ATP that are formed. So we used 2 and we created 4, so in the end we only have created 2 net. And then we also have NAD plus, which is our electron carrier. Remember our bucket that carries our electrons? It grabs some of the electrons and becomes NADH. And then we have some positive protons here as well. And then we have the two pyruvate molecules and some water. So after all of this occurs, after glycolysis is over, we have the energy investment phase and the payoff phase. When we're all said and done, we end up with 2 pyruvate molecules and some water that came from the glucose. We have 4 ATP formed. 2 of them were used initially, so we have a net of 2. And we also have 2 NADH created. And remember, these are kind of like our temporary batteries, kind of holding those electrons, storing that energy till we can move it further down the line. So glycolysis is kind of the first simple step. We're breaking the sugar molecule into 2 smaller molecules called pyruvate. This occurs outside of the mitochondria. From that point, if oxygen is present, we're going to move on into the mitochondria and other processes are going to occur. So concept 9.3 says after pyruvate is oxidized, the citric acid cycle completes the energy-yielding oxidation of organic molecules. So after the pyruvate is oxidized, that happens outside of the mitochondria, that material will then be moved into the mitochondria. Remember, we said it's oxidized, but we said glycolysis doesn't require oxygen. So don't confuse the word oxidation or oxidize with has to have oxygen. Oftentimes oxygen is what does the oxidizing, but just remember that phrase just in general means that the material is losing electrons, becoming more positive. And it doesn't have to be oxygen that causes that, but oftentimes it is. So after the first step of glycolysis, we move on to the second step. We go into the mitochondria. In the presence of oxygen, the pyruvate will go into the mitochondria if we're talking about a eukaryotic cell, and that's where the final oxidation and breakdown of the glucose will happen. If we're not talking about a eukaryotic cell, this is going to happen just out there in the cytosol. So prokaryotic cells that do cellular respiration, they do this process as well, but they don't have a mitochondria. This process just happens out there in the cytosol. So we have those pyruvate molecules coming into the mitochondria. before the citric acid cycle can start, that pyruvate has to be transformed into something else. It's converted into what's called acetyl coenzyme A. Oftentimes you'll see it just as acetyl CoA. That's kind of the nickname or shortened version. And this is basically what connects step 1, glycolysis, with step 2, the citric acid cycle. The process to take the pyruvate and make it into acetyl-CoA is kind of a multi-step process. It takes a number of enzymes. It's actually in 3 little reactions that gets that done. We don't have to know the particulars of each of those 3 little reactions, but we do need to know that the pyruvate is converted into acetyl coenzyme A through a series of enzymatic reactions. So here we have just a quick little brief overview of what's happening. Here we're outside in the cytosol. We're not in the mitochondria. We have the pyruvate here. We have some various transport proteins and enzymes that carries that material across into the mitochondria, and through a series of steps we create what's called acetyl coenzyme A. So we started with glucose. That gets broken down into what's called pyruvate, and then that pyruvate gets changed into what we call acetyl coenzyme A. Now inside the mitochondria we have what's called the citric acid cycle, and it's called a cycle because it's a repeating pattern. We'll see the final product is a material that can be placed back into the system and reused and reproduced again and again. So it's called a cycle because it is cyclical in nature. It's also called the Krebs cycle, named after the scientist most well known with discovering this particular system and clarifying what this system does for us. Sometimes it's also called the tricarboxylic acid cycle. That's a little bit more difficult name, so oftentimes that one's not used. Usually Krebs cycle or citric acid cycle. What this cycle does is it takes that pyruvate, it finishes off the breakdown and basically taking it all the way down to carbon dioxide. So we have the initial sugar, the glucose, that was broken into pyruvate outside of the mitochondria. Now that pyruvate is going to be changed into acetyl coenzyme A, and this acetyl-CoA is going to go into the citric acid cycle. When we're all done with this, what we're going to get from this is 1 ATP, 3 of the temporary batteries, NADH electron carriers, and then 1 another type of electron carrier, kind of another temporary battery called FADH. And that's going to be created per turn. And there's going to be 2 turns of this cycle because, remember, 1 glucose molecule gave us 2 pyruvate molecules. So for every 1 glucose, we got 2 pyruvates. So we'll have 2 turns of the citric acid cycle. Each of those cycles will create these materials that you see right there. Here's kind of an overview diagram. Here is our 3-carbon sugar, or the broken down molecule from the original sugar, the pyruvate. Then we have basically a series of steps to create acetyl coenzyme A. Acetyl coenzyme A enters the citric acid cycle. A wide variety of processes. Luckily, we're not going to need to know every single detail, but we're going to look at a few of the steps along the way. Along the way, we create some NADH, which are like the temporary batteries, electron carriers. We create some carbon dioxide. We create a single ATP, and we create one of these FADH2s, which is another electron carrier, another temporary battery, you could think of it as. And then the material that is created at the end can go back into the cycle and go again. We're going to have 2 turns of this cycle for every single molecule of sugar that we put in, glucose, because we broke that into 2 pyruvates. Now, I want to make clear that you understand that we don't need to memorize every detail of the citric acid cycle. It has 8 steps. Every single one of them is catalyzed by a specific enzyme, and there's a lot going on in here. So I don't want you to get overwhelmed thinking you have to have all of this memorized. I do want you to understand that this is a cyclical process that is happening inside the mitochondria, and for every turn of this cycle, we're creating an ATP molecule, 3 of the NADH electron carriers, and 1 FADH2 electron carrier per turn. So it's called the citric acid cycle because the acetyl coenzyme A is combined to form something called oxaloacetate. Oxaloacetate. Man, try to say that 5 times fast. Oxaloacetate. which forms a material called citrate. And citrate, of course, think about citric acid, the material that you find in oranges and lemons and that type of thing. Through this entire process, there's 7 more steps along the way, but at the end, the citrate is converted back in to the initial material, back into the oxaloacetate. So we end up with kind of what we started with from the first step, and that's what makes this a cycle. So part of the acetyl coenzyme A joins with this material, oxaloacetate, and forms citrate. And then we go through about 7 more steps to decompose the citrate and release some energy and create some NADH. But at the end of the process, the citrate is decomposed, deconstructed back into the initial oxaloacetate. And so that can re-enter the system. That's why we call it a cycle. The NADH and the FADH2 that's produced by the cycle, remember, those are like the temporary batteries. They are relaying electrons that came initially from the glucose or the food, and they're going to take those to what's called the electron transport chain. So they're like the buckets or the vehicles, remember, that are carrying these electrons down the hill. I'm going to show you this whole citric acid cycle. I don't want you to freak out because we don't have to have all of this memorized, but just to kind of show you the different steps. So we have acetyl coenzyme A that comes in. Part of that acetyl coenzyme A gets combined with this oxaloacetate. I'm going to have a hard time saying that in this entire chapter. That forms a material called citrate. Then we have a number of steps. We don't have to memorize all these steps, but we have a number of steps along the way. We have some NADH created. We have an ATP gets created. We have some FADH2 that gets created. Finally, at the end, the material that started off as oxaloacetate and then was made into citrate, it gets broken back down and turned back into the original material to begin with. So we can start that cycle all over again. So again, we don't have to memorize all these steps. Just kind of memorize or understand that it is a cycle that occurs in the mitochondria, and you do need to know what gets generated from every turn of that cycle. All right, so far we've been through step 1, which is glycolysis, step 2, the citric acid cycle, and now we're on to step 3, which is the final part of respiration. So far not too bad. Again, try to keep in mind those big pictures, the big ideas that we're talking about, and try not to get bogged down into every tiny little detail. Just add in as much detail as your mind can hold. So our last concept is 9.4, or the last concept for this portion is 9.4. During oxidative phosphorylation, chemiosmosis couples electron transport to ATP synthesis. My gosh, if you were to read that sentence to your parents, or go to just a store, go to Brookshire's or Walmart and grab the nearest stranger you see and say that sentence to them, there is no way they would understand what you're talking about. But think about it step by step, and I bet by the end of this, that sentence actually does make sense. So it says, during oxidative phosphorylation, so that part, oxidative, tells you it's a redox reaction, which means we're either moving electrons away or toward some material. Phosphorylation, which means we're adding a phosphorus group. This word we don't quite know yet, chemiosmosis, but you know that word osmosis, so that kind of tells you a little bit of what it's talking about. Couples electron transport. We know that this entire process is simply moving electrons around, moving them from a higher energy state to a lower energy state to ATP synthesis. We know ATP is the molecule, the energy carrying molecule for the cells. We know the word synthesis means to create. So this last sentence describes this last step where most of the ATP are created. So in the first 2 steps, glycolysis and the citric acid cycle, we didn't create a lot of ATP. We've only created 4 ATP total in all of the 2 steps, and that's not a lot. So where is the rest of the energy from the glucose? Well, the rest of the energy is still tied up in these 2 electron carriers, NAD+, which became NADH, and FADH2. So these temporary batteries, remember, that we've been talking about carrying these electrons, that's where most of the energy still is. We've created 4 ATP, which is not a lot, but we still have a lot of energy tied up in these electron carriers. These 2 electron carriers are now going to donate those electrons that they've been holding to something called the electron transport chain, and that's going to power the production of ATP via a process called oxidative phosphorylation. This last step, this oxidative phosphorylation and this electron transport chain, this last step happens, again, we're still in the mitochondria. This happens in the inner membrane called the cristae or cristae of the mitochondria. And most of the steps or the little carriers of this whole process are proteins, and they exist in what's called multi-protein complexes. So it's a number of different proteins kind of all working and stuck together that act as a step-by-step chain to move these electrons down toward oxygen, which is the final electron acceptor. So these carriers, they alternate reduced and oxidized states. These carriers are kind of taking electrons, passing them on, taking electrons, passing them on. So in one case, they are taking on electrons, which means they're being reduced, and then they're giving up those electrons, which means they're being oxidized. So they kind of alternate back and forth, and they act kind of as a chain passing these electrons down the line. So as the electrons drop in free energy, as they go down the chain, eventually they're going to get to oxygen, which is their final goal, the electron acceptor, the final electron acceptor, and they're going to form water, which is just a byproduct. It's the byproduct that your body gets rid of through breathing, through sweating, through filtering out by the kidneys, collection in the bladder, and then going to the bathroom. This same process occurs in prokaryotes that don't have mitochondria, but again, it happens out there. Actually, this one happens in the plasma membrane. So in the single-celled creatures, the prokaryotic creatures, this does occur, but it happens in the plasma membrane. In the eukaryotes, we're still talking in the mitochondria. So think of this electron transport chain as a bucket brigade. These electrons, so these are all these various proteins, and they are alternately accepting and donating electrons and moving them down a path. So they are becoming reduced and oxidized. And basically the carriers that we had those electrons in, the buckets, the NADH, passes off those electrons through these proteins. And as these electrons move down the chain, eventually they combine with oxygen to form water. I think of it kind of as a bucket brigade, where in the old days they would take buckets of water. If a barn was on fire and there may not be a fire truck available, or they may not have fire trucks in that little town, they would form a bucket brigade of people that would pass full buckets of water. Someone way up here may be at the well or at the pump filling the bucket initially and then passing it down, or they would try to put out the flames. So this electron transport chain is kind of similar to a bucket brigade. Eventually, we're dropping in energy as we go. That energy that is released, we're going to find out, is used to create ATP. Here's kind of a more realistic view of what we're talking about. So here we have the NADH electron carrier and the FADH2 electron carriers. They're going to drop off those electrons through this big complex system of proteins. But what's going to happen is all along the way they are lowering in energy till eventually they'll reach the final electron carrier, which is the oxygen, kind of the whole target they've been going toward this whole time, and that will create H2O, water. The electrons here of the hydrogen at this point are at their lowest energy state. When they were in glucose, that would be way off the page up here, they were in a much higher energy state, all through glycolysis and then the citric acid cycle, and now down through oxidative phosphorylation and the electron chain. Finally, those electrons are down at their lowest energy state when they're right up against oxygen here. That energy was used, or will be used, to make ATP. So again, these electrons are transferred from the NADH and FADH2. Those were our temporary holders of electrons, our temporary batteries, to the electron transport chain. A long series of proteins, including some cool proteins called cytochromes, which have an iron atom in them. Eventually, we're going to get to oxygen. That's our final destination. This electron transport chain, it doesn't make ATP itself, but what it does is releases energy that goes into a separate machine, cell machine, basically, that makes ATP. So the electron transport chain doesn't create ATP, but it provides the fuel for the next step that we're going to look at. And also this chain, again, breaks this large free energy drop from food to oxygen. So we don't have this explosive release of energy all at once. We have this step-by-step process from the glucose all the way to the oxygen, where energy is released in small, usable amounts. So finally, last but not least, what are we releasing this energy for? What's this little cell machine that I keep talking about that's going to create these ATP? Well, this process is called chemiosmosis, and it's called an energy coupling mechanism. So the electron transfer in this whole transport chain causes protons to pump H plus ions from the mitochondrial matrix to the inner membrane space. So all of these little proteins that were passing the electrons down the chain, as they pass those electrons down the chain, that changes these proteins and causes them to pump H plus ions into the mitochondrial matrix. And we're going to find out that's going against the gradient. That's going against the concentration gradient. So they're using energy as these electrons pass through them to pump H plus ions into this mitochondrial matrix, from the mitochondrial matrix into the inner membrane space. Now those H plus ions are building up in concentration in the inner membrane space, and they would rather go back across the membrane and even out that charge. Remember we talked about osmosis and the electrochemical gradient. They want to even out that charge, so they want to diffuse across the membrane and get back to where they were, or get back to the other side so they can even that out. As they diffuse across, they pass through something called ATP synthase. Oh, I just noted a mistake. This said proton. It should say protein, so I fixed it to say protein. As those H plus ions, as they try to get across that membrane to even out that whole electrochemical gradient, they're going to pass through a protein called ATP synthase. ATP synthase is the machine that creates ATP. It's the protein that does this work. It uses this flow of H plus ions to drive what's called the phosphorylation of ATP. So this is what we call chemiosmosis. It's an example of chemiosmosis. We're using the energy of the H plus ion gradient to do actual work. So what we're saying is the energy that gets released through the electron transport chain is used for active transport to move H plus ions against the electrochemical concentration gradient. And then as those H plus ions flow back to where they would naturally want to be, that flow is what powers ATP synthase. So let's take a look, because a picture is always more helpful than just words. Okay, so we're inside the mitochondria. We have the inner membrane space up here and the mitochondrial matrix area here. Here we have this protein called ATP synthase, and it almost looks and acts like an actual little machine. So the other proteins, the electron transport chain proteins, we're using those flowing electrons to pump H plus ions into this space in the inner membrane. So there is a lot of H plus ions up here in the inner membrane space, way more H plus ions up here than there are down here in the mitochondrial matrix. They want to even that out, so a lot of these ions are trying to diffuse across this membrane. So instead of just going directly across the membrane, they're going to go through this particular protein called ATP synthase. As those H plus ions travel through ATP synthase, that actually changes the protein and causes the protein to almost move like a gear. That motion and that process allows the protein to take an ADP molecule, which is adenine diphosphate, and add to it a phosphorus group. That creates, ta-da, ATP, adenine triphosphate. So this machine, almost like a cellular machine, is powered by H plus ions flowing through it. And that machinery combines a phosphorus group with ADP to create an ATP molecule. So think of it kind of as a water wheel, like perhaps you've seen in old movies or pictures where they use a running river or stream to power a paddle wheel of some kind, and then that does some work inside the shed. Well, here you have a machine powered by the flow of H plus ions to create ATP. So here's an overview of this final step that we're talking about. We have NADH and FADH2 dropping off these electrons through this long chain of proteins. As these electrons move down the path toward oxygen, they are powering the pumping of H plus ions into this space. Now there's a lot of H plus ions already up here, so this is against the natural electrochemical gradient. We are adding a lot more positive charge, and we're adding a lot more H plus ions up here. Well, those H plus ions would rather just come across this membrane and even out the electrical charge and even out the whole gradient. So as they move through this protein called ATP synthase. This protein is like a little machine. The power of them traveling through allows that little machine to phosphorylate ADP. In other words, add another phosphorus group, and that creates ATP, adenine triphosphate. So this part is the electron transport chain. This part is the chemiosmosis. All together, that's called oxidative phosphorylation. The oxidative part means we have redox reactions going on. The phosphorylation is the addition of a phosphorus group to ADP, creating ATP. The energy stored in that H plus gradient that's what powers this final step to make ATP. So this H plus gradient, in other words, this concentration of H plus ions on one side of the membrane and flowing across to the other, that's called a proton motive force. Because of that higher concentration on one side and the fact that it naturally wants to flow across and even out that gradient, we can actually do work with this flow. And so it's called a proton motive force. And this ATP synthase, a little factory, this little machine, it's what produces most of the ATP in this whole process. It can make up to 28 ATP molecules. So let's do a final accounting of what we've done, what we've created. So in cellular respiration, most of our energy looks like and flows like this. We start off with glucose, and we're just going to look at the energy flow. So where does the energy go? So we have glucose, and then most of the energy from there goes into NADH molecules, then into the electron transport chain, into the proton motive force, and then finally into ATP. So most of our energy in this whole process goes from glucose to our little electron carriers, finally to the transport chain. That helps create that proton motive force, and then into ATP. About 34% of the energy in a glucose molecule is transferred to ATP, making up to about 32 ATP. So 34% is not very efficient. So a huge amount of energy that's in the glucose molecule is used to power this whole process, or is lost along the way as heat and other sources. So only about 34% of the energy in a glucose molecule ends up as ATP, useful energy for the cell. But it's still a process that works. Apparently it's worked for quite some time, but I wouldn't call it the most efficient process. So why do we keep saying about 32 ATP? Why isn't it exactly 32 ATP? Well, there's a couple of different reasons. They have to do with the inefficiency of the entire system itself. They have to do with the fact that there can be different electron transport mechanisms. Some of them are not quite as efficient. And also they have to do with the fact that the cell uses that proton motive force, the concentration of H plus ions on one side of the membrane, to do other work as well. So not all of the energy from the proton motive force goes to making ATP. The cell also uses that for other reasons. So we can make up to about 32 ATP if all of that energy goes toward that one particular process. So let's look at the whole process overall. This is probably a slide that you want to try to kind of burn into your memory just a little bit. So we started off with glucose molecules outside of the mitochondria. Out here in the cell cytoplasm area, we have what we call glycolysis. Glycolysis breaks glucose into two pyruvate molecules. In that process, we create two ATPs, net two ATPs. We actually had to use two to start with. We created four, but we netted two ATPs. We also created 2 NADHs carrying electrons, kind of like our temporary batteries. Then we have, if we have oxygen, we move across into the mitochondria where we have pyruvate oxidation. That's going to take that pyruvate and further change it to acetyl coenzyme A. These electrons were carried across, and we have NADH, FADH2. We have 2 more NADHs created here, so we have more energy stored there. That acetyl coenzyme A goes into the citric acid cycle, and that basically is 2 turns because we have 2 pyruvate molecules to start with. That's going to create 2 ATP as well. That also created some more NADH and FADH2s. Finally, we get to the last portion here, oxidative phosphorylation and the electron transport chain and the chemiosmosis. The NADH and FADH release those electrons to fall down that electron transport chain. Sorry, I can't talk. That pumps H plus ions across the membrane, which then, as they flow back, ATP synthase uses that flow to create somewhere between about 26 or 28 ATPs. So, total, altogether, we have usually about 30 to 32 ATP from one molecule of glucose. All right, we're going to take a pause right there. I know this is heavy material. I know this makes your brain hurt. But again, let's break it down into these 3 parts and think about what's going on. Think about the fact that electrons, usually combined with their little H+, so we're talking about hydrogen atoms here. Electrons, the hydrogen atoms, are stripped from the glucose, and they're basically going through this entire complicated process to move those electrons of the hydrogen atoms down to lower and lower and lower energy states. And as they move down to lower energy states, the energy that is released is used to basically power the creation of ATP in the end. So along the way, we create a few ATP, but most of that energy is stored in these temporary storage containers or buckets or batteries, which then finally gets released through the electron transport chain, which pumps H plus ions across to one part of the membrane. Then as they flow back through diffusion, that allows the ATP synthase to crank its little machine and create the large amount of ATP at the end. So try to think big picture. Don't get too bogged down into the tiny, tiny details. Remember, we don't have to know every single step of the citric acid cycle or every single step along the way. I'd rather you focus on the big ideas, and you'll do better that way. We'll take a break. We'll be back with fermentation and anaerobic respiration, and we'll finish up.